did this chapter quickly, right?
Yes, right. But what I can offer you is, when is your exam?
My exam for biology is on the 10th of November, sir.
10th of November. Okay, so I'm starting biotechnology in like in another batch. So you can join that class if it permits your schedule.
So like which all days?
I'll let you know. Are you on the group? I'll send a
Yes,
message
sir, I'm
to
on
the
the
group for the biotechnology class, the timings. And anyone who wants to just study that parallelly can just join that, okay? Because I think only the first chapter will be coming, biotechnology, the fundamentals.
Introduction.
Introduction, not the application, right?
Right, sir.
Yeah, okay. So we'll sort that out, okay? No worries. Just 1 second. Hello. Yes, am I audible, everyone? Hello, am I audible?
Yes, sir.
Yeah, okay. So today will be, yes, anyone has any other doubts? Fatima, when was your exam? Is it done?
It was the 19th.
It's done, right? How was it?
It was good, but then there was one question which I had no clue about. There were 3 scientists and they asked what was their contribution in genetics.
In genetics. Oh yeah, that's one thing that's one weird thing. I don't know what to say, but there's one thing that school exams do. So when you read NCERT, you will see certain scientists' name and their contribution, right? So they expect you to know that. And for some, it's easier because they have made significant contributions, which while reading, we understand. But what were these 3 scientists which you were clueless about? And
I have to check 1 minute. I'll just be back.
Yeah. Because in genetics, there are just handful, Morgan, Mendel. And if these 2 are not there, then Bateson, Francis Crick. These are the ones.
It can also be Avery, MacLeod, and Mockhart.
Avery, MacLeod. So, but these 2 we studied, no? These 2 we studied as experiments, right?
Oh yeah.
But sometimes they even ask you, like, for example, when plant begins, there's this a small intro about Sir Pancham Maheshwari. So we don't study about him in the chapter, but yeah. What? Who are
It
they?
is Hargobind, Farana, Marshall, Nerenberg, and Severo.
So that's like in DNA fingerprinting, I think.
Yes, sir.
Yes, right. So that's towards the tail end, and they give you these short snippets of various scientists. Hargobind Singh Khurana was very, very, very famous Indian scientist, molecular geneticist. So they expect you to know these things. So when, like, for example, in plants, I was telling about Pancham Maheshwari. So Pancham Maheshwari's short bio is there. So just do read that, because no one can know which scientist they can ask. But apart from that, any other thing? I don't think you should worry about that question. If you don't know scientist contribution, it's fine. Any other logical or conceptual thing that you missed? No? Okay, cool. So since you have given your examination, I will, how about Arpit, how about you giving a summary today?
Yes, sir.
Yes, we started in the last class with pneumonia, right?
Sir,
No.
but like I also have a lot of disturbance because I'm with my family.
It's okay.
That's
It's completely fine. We all are at home, so we all understand that. It's not something very, very board meeting kind of a thing. And these days, believe me, board meetings are also happening from their couches and sofas, so it's okay.
yes. So we started with malaria in the last class. So we discussed about the causative agents of malaria. It is caused by Plasmodium malaria, Plasmodium vivax, Plasmodium ovale, and Plasmodium falciparum. So the mechanism is it is a two-host system, and the process is like the mosquito, it bites the normal human, then the parasite, which is known as the sporozoite, it enters and in the blood and it reaches the liver, In the liver, it first starts replicating, and
Mhm.
asexually it produces asexually, and then after some time it bursts and it enters into the blood. Now they also reproduce in the RBC, and later they burst, which leads to the causing of symptoms such as fever, and it also releases a toxin which is known as hemozoin. And the system is happening in the human host. Later this also forms gametocytes. Now the next part is happening in the mosquito host. When mosquito bites the infected person, now the fertilization happens inside the mosquito gut, and it gives rise to sporozoite, and then sporozoite are stored in the salivary glands, and the cycle continues. So then we did the symptoms. Symptoms are fever,
Yeah,
flu.
yeah. Before you go forward, just one thing I want to add. Very like a very detailed and brilliant explanation. But so you said they also produce toxin hemozoin. Actually, toxin hemozoin is the central reason for these chills and the symptoms. Okay, it's not something which is just produced as a byproduct. This is the thing which causes this chills and fever and the whole cascade of response in inflammatory response in the body. Okay, so
Yes.
whenever sporozoites burst and come out from the RBC in very large numbers, these this toxin also increases in the blood because that is also released. Yes, continue.
Yes, sir. So the symptoms for this is like we get fever, flu, shaking, chills, headache, some muscle ache, vomiting, etc. So within the amoebiasis, sir, and it is caused by Entamoeba histolica. And sir, the mechanism is like they attack on the large intestine, and the carrier for this is the housefly. The symptom is it causes diarrhea. There's pain in abdomen region. There is sustained fever, headache, and cramps. The mode of infection for this is contaminated food and water, and basically it's through fecal route. And so
Yep.
then you told us to do the Escherichiasis and Elephantiasis on our own. So
Did you do that?
yes, sir, I
Everyone?
did.
Okay, anything you found difficult or
No,
you
sir.
could not understand? I think it's very plain if you follow the same route, like the same plan. You just have to understand what causes it, what is the mechanism, what is the organ that is infected or affected, and something specific if the symptoms are something specific. Okay, it's all it's simple, right?
Yes, sir.
Okay, so let's begin with some important things today, some concepts which are going to help you later on also. So immunity. So we discussed immunity. We started with immunity in the last class, where we understood that immunity is the overall ability of the host system to defend itself against the disease causing pathogens, right? Now, when we say immunity, what is it? Like this overall ability, is it some kind of a superpower? How, do you think it makes sense on a molecular or a cellular level also? What do you understand by immunity? And I told you there are two types, innate and adaptive. Anyone familiar with these words?
Yeah, innate immunity is the immunity that you already have since like birth. And adaptive immunity is what you like develop throughout time, like getting exposed to like more like pathogens. You like develop an immunity.
Okay, so if I write it, what Matthew is saying, innate immunity is by birth. So can I say it's kind of a default setting in our system,
Yeah, you could say so.
right? So anything that you buy, any gadget that you buy, it comes with some default settings, right? Right from when it comes, yes, Arpit.
No, so first you complete, then I'll
Yeah.
ask.
So any gadget that comes, I was saying that it comes with some default settings, and then we tweak with certain things depending on what kind of performance we want from that thing. Similarly, innate immunity is by birth. So which means organisms have evolved over time. Like we have studied evolution. Now you have a context of evolution. So when I use the word evolved, you know what's happening. So organisms have evolved competing with each other. And when I say organisms, it includes unicellular organisms, multicellular organisms, all plants, animals, birds together. So competition is one thing that has happened in nature since the beginning, right? We have outcompeted other species of the genus Homo. And that's why we Homo sapiens are the only species staying. But when it comes to a smaller organism, which can be unicellular and can act as a pathogen to a multicellular organism. So what the smaller organism wants is to somehow get into the system where it can multiply, get food, hijack the host system, and enjoy. By enjoy, I means live the cycle, make more replicates. And what the host wants, it's exactly opposite, to not let that happen. Correct?
Yes,
Makes
sir.
sense?
Yes.
Yeah. So this thing, when unicellular organisms enter the system, multicellular organisms also detect them. So they have a detection system, and it's very important. If you do not have any detection system, if you do not have patrolling police in the city, petty criminals like theft, burglars, they will just do whatever they want, because they are not, when they enter these organisms, they do not come with a pomp and show. You will not see them coming. For example, COVID is very sneaky, right? You will not know when you get infected. It just takes 10 to 15 minutes of exposure and you are COVID positive. But once it is inside the body and it starts replicating, you should have a system that can detect it. That gives rise to immunity. So over the time, over the course of evolution, all the organisms that can be hosts, and believe me, every organism is hosts to some other organism. Okay, so bacteria are pathogen for us, but there are bacteriophages which are pathogens for bacteria. So a smaller particle can be pathogenic to a bigger particle, but it is again, bacteria is again one single or single celled organism, right? So innate is by birth, which is hardwired in our genes. So we pass on this information. Mother passes on that information to the baby that, see, you need to have these things in your system right from the birth, because it's going to help you. But what mother does not know is the future. What's going to happen? Which kind of pathogens are going to emerge later on, correct? Because before 2019, none of us know what's COVID, what COVID can do, right? The novel COVID, SARS-2 can do, or where it attacks. We only know about it now, right?
Yes, sir.
So we could never have a immunity against it. So many things develop after birth, because new kind of pathogens, you are exposed to something which your parents were not exposed to. So clearly you develop something. You develop new programs in your system. That's adaptive immunity. Yes, before I move forward, I'll take Arpit's question. Arpit, what were you asking?
Sir, I was asking, like, is adaptive immunity and acquired immunity same?
Yes, they're same. There are two words. Adaptive immunity is also known as acquired immunity because it's acquired after birth. Innate immunity is also called neonatal, by birth. Okay, or the it's also called, it has one more name. Okay, I'll remember it, I'll tell you. Okay, so it's by birth, it's acquired. Now let's go into detail of innate immunity a little bit. So first question first. So you don't have to remember. I'll try to make immunity so simple to you that you will not need to go and mug it up if you just pay attention to this class. Now innate, by birth, like a program which is preloaded in a system. And this program has one job to do, to detect the pathogens, trace the pathogens, kill the pathogens, right? Immunity has just one thing to do, correct?
Yes, sir.
Yes.
Now, if a type of immunity is already given to us right from birth, what do you think? What should be the mode of function or extent of that immunity? Because you cannot make it specific. Can you make it specific to something? Like, can I make a innate immunity specific to tuberculosis bacteria or COVID virus?
No.
No, sir.
No, right? Because that level of specificity can only come when you are, that also happens. Like for example, bacteria, bacteria have some specific type of immunity against some bacteriophages, and that whole system, that defense mechanism is called CRISPR. Anyone has heard of the term CRISPR technique? It's a gene editing technique that we have utilized for our benefit, but we have taken it from bacteria. So bacteria use it for its own defense against bacteriophages. So that can only happen when for millions of years, these two things are constantly competing with each other, or trying to, attack each other or defend oneself from the other. But innate immunity is normally generalized. It's like, to give you an analogy, if you I don't know if people are familiar, but Windows used to come with a firewall. It still comes with a firewall. No one uses that, but it's called Windows firewall. It's a very generalized way to protect your PC against threats, but it cannot pinpoint what kind of threat, a Trojan or a virus or a malware, what kind of a threat it is in your system. But it's a basic thing. Like what you can do is, if anything seems malicious to the system will not open it. It will show you a warning and will not open it. But all the time it may or may not be harmful. Most of the time it may be harmful, but you will not know what harm it can cause, because it's just general. It will say ki, don't do this. Similarly, innate immunity have some very basic players that cannot distinguish between different types of pathogens per se, but can treat all pathogens that they can identify same. They just can try to degrade it, and most of the time they are successful also. And innate immunity evolved earlier. Like, why do we say that? Because invertebrates have only innate immunity. They don't have acquired immunity. Invertebrates like your house fly, your other flies, dragonflies, butterflies, what else? Earthworm. All these are invertebrates. Okay. Anything apart from chordate phylum are invertebrates. They don't have adaptive immunity. They have innate immunity majorly. So simply, innate immunity is non-specific. If I say, so you understand what is non-specific? It's by birth, non-specific, and it basically is like kind of a barrier. Barrier system. Now, a barrier is just like a wall, correct, everyone? A wall does not distinguishes between who's trying to break it. Make sense? Just give me 1 second. Hello, am I audible, people?
Yes, sir.
Yes.
Yeah, so I was saying, if something is a barrier, that barrier will not know, what kind of thing is trying to penetrate it. Similarly, innate immunity, so this barrier can be at 4 levels. Everything that you have by birth can participate in innate immunity. It does not have to be just the molecules or specialized cells in the system. It can be something as trivial as your own skin, starting from the skin. So let's go from outside to inside, and let's see what all are part of your innate immunity. So innate immunity acts at 4 levels. The first one, which is the outermost, is called the physical barrier. Okay. Now, as the name suggests, it's just a physical barrier, just to hold things out. And can you tell me what all things can come in this?
Skin.
Skin, for sure. How do you think skin acts as a barrier? Just the logic, anyone? Why does skin act? Why is skin a very good barrier? Anyone knows? Yeah, yes.
So it does not allow any pathogen to enter.
But pathogen can enter in the skin cell itself, no? Just like a pathogen can enter in our lung cell, in our throat cell, eyes. Why can't pathogen enter in the skin cell from outside? Why can't anything enter just from the outside into a tree trunk? Because the layers on the top are dead. Simple answer. Our skin, multiple layers at the top is dead. It's just dead layer. Living skin starts multiple layers below. Okay? You understand, people? So any pathogen, any bacteria, virus sitting on your skin is harmless. So in case of COVID also, if the virus is on your skin, you do not have any issue. It should not just enter in your nose, because there inside you have living epithelial layer where they can penetrate and get inside the cells. So skin is dead tissue on the outside. Some few first initial layers are dead. And you can also test it. If anyone of you during the winters have witnessed this, that if you just get something, nail or something over your skin, there is a white line that you see.
Yes, sir.
That's if you make that white line and see it under the microscope, you will see that those are dead skin cells. Okay, so that's how skin acts as a good physical barrier. And our openings, all the openings of our body, nose, eyes, ear, mouth, all these are where the skin ends, like, and there is an opening inside. So living tissue is exposed to the outside. Those are the entry points for a pathogen also. Okay, not the skin. So skin is the main barrier which prevents entry of microorganism. Main barrier. Okay. Prevents entry. Of microbes. Then apart from skin, anything else can you think of which can come in a physical barrier? Where there is no skin, there is mucus. You know what is mucus, everyone? Mucus.
So hair can also be like a barrier, no?
For immunity, you are saying, right? Hair can be a protective layer from the environment, from the outside environment. But how do you think they are preventing pathogens to enter? Or you are talking about the hair inside the nasal cavity?
No, I was talking about
Body
normal.
hair.
Yeah.
Yeah, right. So hair basically have multiple roles in biology. Hair is not just small hair-like things on your skin. Also helps you in touch sensation. Sometimes something, if it comes very close to your skin but still have not touched your skin, you can still sense it. Try taking the pen very close to your skin, very close, but don't touch your skin. You'll still feel, because some hair-like particles, you will touch the pen to those small fine hairs, and they at their base have these sensory receptors, touch receptors or stress receptors. But here we mostly say that they are insulating. Humans have lost a lot of hair from their body during evolution because we started wearing clothes. But many other mammals, for example, our closest cousin in evolution, chimpanzees, they still have their full body covered with hair, right? Because they are not wearing clothes. Yeah, and that's also true for other non-primate mammals like rat, goats, even buffalo, cows, etc. So, but after skin, if something really acts as a barrier for pathogens, is mucus. So all your openings, all our openings have mucus. In fact, all the internal tracts are lubricated with mucus. So nasal cavity, oral cavity, the buccal cavity, esophagus, food pipe, windpipe, intestines, stomach, all are heavily lined with mucus. So what does mucus do? Write down, mucus coats the lining coats the lining of all the tracts in our body. All the tracts. Now you know the tracts. They are respiratory tract, gastrointestinal tract, reproductive tract, urogenital tract, all these. All the tract, and trap microbes in it. So if anything tries to get through our nose, the mucus should protect us, but cannot protect completely. And that's the reason we get COVID, clearly. Anyways, but skin and mucus, yes, they are a part of innate immunity as physical barriers. Second, let's go one layer inside. From the physical barrier, let's go to physiological barriers. Now, what do we mean by physiological barriers? What is the difference between physical and physiological word? Anyone knows? Physical and physiological. Physiological is what is present inside the body, okay? And also have a relevance in our functioning. You understand? For example, if bacteria, viruses, or pathogens can somehow fool or go beyond the physical barrier, which they normally do, okay? If they are very contagious pathogens, they normally do. Normally, our skin and mucus protect us from millions and millions of virus and bacterial and fungal particles on a daily basis. But sometimes some contagious ones can just go beyond this. Now, the next place they can go is the physiological barrier, which is, first is acid. Acid in our stomach is a very good physiological barrier because it just destroys anything that comes into it. For that, given that you are producing acid in a normal quantity, if you are not producing enough acid, then this barrier can also fail. But acid in stomach is a good barrier. And similarly, there is something called saliva in mouth. Now, if you know the pH, acid itself, in our stomach, we have a very strong acid having a pH of around 1 to 2. It's a very, very strong acid. It's as strong as,
How is it not harmful for our body?
Exactly, beautiful. A similar question I can ask you: why snake itself does not get killed with the venom that's storing inside its own body?
Yeah.
Have you thought about it? Biology is fascinating, right?
Are they resistant to that?
So they cannot be resistant to that because they are also following normal norms of biology. Yes, you can be resistant to toxins over time, but that's not what's happening here. Because if they're resistant, then if you inject a cobra's venom into the cobra's body, like with the injection, cobra, there should be no response, right? But snakes do die from their own venom, which means there's something that's happening here. When we produced, anyone knows how does our stomach produce acids? And I have given you this answer partially multiple times, I remember. Any acid in our body, any biological acid, in what form does it stays in the body? Okay, tell me which acid is present in the stomach?
Hydrochloric acid.
Hydrochloric acid, good. HCl. What is HCl? H is hydrogen and Cl is chlorine, okay? H plus and Cl negative, right? These two together form HCl, correct?
Yes, sir.
This form HCl is an acid. It's corrosive in nature. But H plus and Cl minus alone, separate, are not at all harmful. They are actually just ions, one among the thousands of different ions present in the cell. So the cells which produce these acids and secrete it in the stomach do not produce and keep it as HCl in the cell. There are cells that produce only H plus, and there are cells that produce only Cl. So they release these ions into the stomach lumen. Now, once you release it in the lumen, they will make chemical bond and form HCl, but then stomach is heavily lined by mucus. You know that?
Yeah.
And mucus is, you cannot corrode mucus with acid. You know, you cannot store acid in metal containers,
Yes,
but you
sir.
can very easily store it in a plastic or a glass container, right? So it's not about the strength, it's about the compound. Mucus is inert to acids. So our stomach has a very thick lining of mucus everywhere. So when H plus and Cl minus are released in the lumen, where they combine to form HCl, our stomach is still safe. The stomach lining is safe. Do you understand now, Fatima? How are we?
Yes, sir.
But what you asked actually happens sometimes, where people who cannot produce enough mucus in the stomach, now they undergo a problem. If you cannot produce enough acid, if you cannot produce enough mucus,
Sir, it
or-
causes ulcer.
yes, then what happens is you cannot coat the complete gastric lining, and the HCl starts killing corroding your own stomach cells, and those are called gastric ulcers. Gastric means stomach and ulcers means wound kind of things. So gastric ulcers happen.
Sir,
Or yes,
no, sorry, first.
no, I was just saying, or the other way it could happen is you are producing normal amount of mucus, but you are producing more than required acid, which sometimes happens in response to medications. So if you have noticed, all of you, whenever you go to a doctor and doctor gives you something like antibiotics or any drug which is potent, they always give you an antacid with it. Pandol or Pandy, they always give you that. It's to keep your acid secretion regulated in the stomach. And you always take it before the medicine, right? They always say that first take Pandy, then after some time take the medicines. Or mostly people take it together, but ideally you should take it before the medicine, because that will go and regulate the acid secretion, and then your medicines, whatever drug it's going into your stomach, will not cause hyper acid secretion. Yes, Arpit, your question.
Sir, I had a doubt. Sir, we know that the dysentery, sir, it is caused by a bacteria, and sir, it affects the small intestine. So, sir, going to the small intestine, sir, it also have to pass through the stomach. So, sir, does not the HCl kill that bacteria?
Good question. Very good question. You know, bacteria is also evolving over time, right? One thing is your concentration. If you are anything that we eat, we can never eat uncontaminated, bacteria proof or pathogen proof food. They are everywhere around us. Remember, I told you that if you just rub your finger on any table or any surface of your home, there'll be millions and millions of viruses and bacteria and fungal spores sitting on your finger. But the point is, they are all not attacking humans. They are not, human is not their host, so not a problem. Even sometimes the pathogens which are harmful, if their number is not that much, not that high, then also it's okay. They go into the stomach and they stay in the stomach, and our acid can kill it. But sometimes you go and eat from roadside and you get the food poisoning the next day, right, Harpit? I think that will also. That happens because there is too much of contamination in that food. The contamination load is beyond our stomach acid can handle. Or when we eat food and we don't chew it properly, and if we are eating food with a bulk of pathogenic load, before our acid can kill all of them, they pass on into the intestine, because there is a limit till which 4 to 6 hours food stays in the stomach. That's all. It cannot stay in the stomach forever or for 12 hours. Howsoever amount of food you eat two sandwiches or you eat 8 sandwiches, they'll stay in your stomach for a time. Then they will pass on to the intestine. So that's why overeating is not good. It will not give you beneficial things often. And the question that you asked, the another answer to that could be some bacteria under unfavorable conditions undergo into cyst-like stage, where they make a very hard outer covering around them. They shrink and they look like desiccated, dried up, and there is a very hard covering around them which forms. Now this protects them from even acid in the stomach. And when they again go into the intestine, where again this condition is alkaline, it's favorable, they come out of the cysts and start multiplying. Okay.
Yes, sir.
Yes, I think Kulsum has a question in the chat. She has written, what stimulates the acid production? What initiates it? Yes, first thing is your food. So you have a rhythmicity. Our system, we have a circadian rhythm set. If you eat breakfast at a particular time, every time before that breakfast, like if it's a habit, if you have a habit of eating breakfast at 8 in the morning every day, then you will realize that from 7:30 onwards, your body will start producing saliva and acid in the stomach, because the body knows that the food is about to come. So it's making preparations for the oncoming food. And if you don't eat, like you all must have observed it when we observe fasts. So when you are in a habit of eating, then the first fast comes, right? And observing fast is there in every culture. If you have observed fast, if you have not observed fast and because of some work you skipped a lunch, let's say you were very regular with your lunch and one day you skipped it. So what happens is you start feeling that something is bubbling in your stomach, right? Do you feel that?
Yeah.
Even stomach make noises, which because the acid is produced, but you did not give anything to the stomach to break down. So now the acid is just bubbling inside. Okay. And if you keep observing fast every day, then again it takes some time, a week or so, for the body to set into the new settings. Okay, now I should not expect food because this person is not going to give me food. So the stomach sets, acid production decreases over time. And then it's a very cool experiment you can do on yourself, your own physiology. The fasting period ends and the next day you are on a feast. From fast to feast, then also it becomes, you get indigestion the first day, or constipation. Has it happened? Or loose motions? Any three of this, because you will not be able to digest your food properly because you have not been eating lunch for last weeks, and then all of a sudden you started eating lunch at the same time. So body again takes some time to set in and start producing acid. Do you understand?
Yes.
Everyone? That's called internal homeostasis to things. Other things that can, this was part of the answer, okay, Kulsum. Other things that can stimulate acid production are these medicines, these drugs, okay. That's why the drugs which are more potent, they always give you antacids along with that. The doctors give, and you should take it. It protects your stomach. Okay, so acid in the stomach and saliva in the mouth are physiological barriers. Can you think of any other physiological barrier apart from these two? There's one more thing which protects our eyes. It's called tears. Tears are not just an, expression of your emotion of being too happy or too sad. Tears actually are biologically and physiologically, they're slightly acidic. If you think, that's why if someone has cried for a very long time, the eyes get puffy and red. They seem like an allergic response. Why does the eyes get puffy and red? Because it gets irritated by the constant secretion of tears. But if something gets into your eye, something very small, your eyes will instantly start releasing tears. Yes? Yes or no?
Yes,
Yeah.
sir.
In some humans, there's a disorder where you cannot form tear. Now, that is a very, very critical situation because your eyes just dry off. So those people always keep these lubricating thing and they have to keep adding drops to their eyes after every some, I think hours. So multiple times in a day, you have to lubricate your eyes. So tears keeps your eyes wet, protect them from drying, and also kills any pathogen that tries to enter through the eyes. So tears in eyes are also physiological barriers. Okay.
Sir, what if pathogen try to enter from ear?
In ear, we have wax, ear wax. So ear wax is majorly for, it's a waste from the ear that keeps the eardrum clean or keeps the eardrum lubricated. But yes, if it tries to enter through ears, then there is ear wax. But pathogens do enter through ears and nose and even through eyes. You all studied amoeba, right? Did I tell you about amoebic dysentery? This Amoeba histolytica can also enter without a vector, directly from your nose, ears, or eyes. If you are taking a bath in an infected lake where this Amoeba histolytica is there, so it can just enter through your nose, eyes, and ears.
Yeah, you told.
Yeah, and it was such a case was reported recently, where this Amoeba was eating up this kid's brain, and then he got to know this. Okay, so all these three do not allow or prevent the microbial growth. That's why they come in the physiological barrier. Now let's go into a little more deeper in biology. So physical, physiological. Now let's go to the cellular level. So then there are cellular barriers. Is it making sense, everyone? It's not difficult, right? We're just taking a tour as a pathogen from outside to in. So if, let's say, there's some very strong pathogen that somehow surpassed the physical barrier and also surpassed the physiological barrier, and now is inside. For example, this dysentery which you were saying, right, Arpit? Now it has happened. It surpassed
sir.
all the barriers. Now what? So what do we mean by cellular barriers? Cellular. There are cells now in the body which are responsible. So these cells which are present by birth and a part of innate immunity, cellular barrier of innate immunity, are called leukocytes. You all know about it. You know what are leukocytes? They are called WBCs, white blood cells. Now in white blood cells, we have many types, but the most important that play a role in innate immunity are neutrophils. Let me tell you that leukocytes will also play an important role in adaptive immunity. Okay, so you have to remember which play the role where. So neutrophils, they neutralize things. That's why they are called neutrophils. Monocytes, and NKCs, natural killer cells. These are the three types of WBCs. These cells, NKCs, are programmed to just kill pathogens or any foreign particles. That's why they are called natural killer cells. NKCs, they are right from the beginning in our system. So they are present in the blood. All these three are present in blood and are a part of cellular barrier of innate immunity. Apart from these, there in the tissue also there is something, a component of innate immunity, which is called macrophages. Now these cells, macro means big, they are big cells. They're again WBC lineage, but they just eat up anything that is not part of our own body, that is foreign particle. They will just identify it and engulf it. Literally, they do that. Just like amoeba engulfs. You all must have drawn this diagram of amoeba feeding, right? It extends the pseudopodia, engulfs the food particle, and put
Yes,
it into a digestive vacuole and digests it with enzymes, right? Similar kind of thing macrophages do. And if you go to YouTube and you see the real videos of macrophages, literally time-lapse videos. So you will see these macrophages in culture mediums. When you put a bacteria, they literally chase the bacteria and then engulf it and destroy it. Okay. So they literally are just phagy means eating, macro means big cells that eat up other kind of cells. So they are in the tissue and these three are in the blood, and together they make the cellular barrier of the innate immune system. Is it clear?
yes, sir.
Okay. Now leukocytes will come again. So but just remember. Let's say something now has even surpassed the cellular barrier. Neither macrophages nor these leukocytes, no one could see that virus, and that virus, let's say, entered a cell. So it has evaded everything and it has successfully infected a cell. Now can something be done, or is the battle lost now? Because biology has been evolving from millions.
Antibodies can be made.
Antibodies are a part of acquired immunity. Fatima, you're correct, that happens, but that's not a part of innate immunity, because antibodies are specific, right? You can make antibodies against specific pathogens. That's where the difference is. Think about from an innate perspective. My question is, do you think that a system or a human or any organism can be born with something that will not just give up at the cellular level, and even if the virus infects the cell successfully, there is still some component of innate immunity that can work? Because it's very important. I told you that all invertebrates have just innate immunity. That's important. And there is something which is called the cytokine barrier, the last barrier in innate immunity. So we are going inside the cell now. Cellular barrier, the word cyto means? Cyto, the word cyto.
Cytoplasm.
The word cyto means cytoplasm. What in cytoplasm also there is a word cyto. This was the first word which I made you write in your terminology section, I think.
Cell.
Yes. The word cyto means cell, cytology, study of cell. Cytoplasm, the fluid of cell. Okay, cytokinesis, the division, kinesis, breakdown of cell. Cytokine barrier. So these are, it is also known as the molecular barrier, because now we have entered inside the cell, and inside the cell there are things, proteins called interferons. Interferons. Okay, so these interferons are proteins which are secreted from inside those cells which are infected. Let's say there is a virus infected cell. Now the cell which is infected knows that I cannot survive. I will for sure die because the virus has hijacked my machinery and it's making its multiple copies, millions and millions of copies. Now it will kill me.
It will warn the other cells.
Exactly. So interferons are a warning system for other cells, non-infected cells, to protect them from the further infection. Now, how beautiful is that in biology? Now, this is something where the cell is not looking for its own survival, but the system is looking for its survival at the cost of sacrifice of that cell. So this cell starts secreting interferons. Write down. A virus infected cell secretes interferons, which warn and protect. Warning, which warn and protect non-infected cells from further viral infection. But then viruses are very, very sneaky and evolving fast. They've also figured out how to surpass this. Because if this is the case, if one cell who gets infected first can warn and protect other cells, then technically we are sorted, no? We will never have any viral infection. Why did we have COVID then, right? Because viruses sometimes can go very sneaky. They go
They're very adaptive.
Yes. They even do not let the cell know until the time until its own death. Okay. So that's the catch. But this is the limit for our innate system. Our innate system can only go into this much of detail and depth. And if I ask you, do you think innate system is outdated? It's not good enough. What do you think? Is innate system good, or is it not good enough?
It does help us, but not completely.
So what should I say? Like, it is like, is it important to have innate immunity or not?
Yeah, it is important.
Yeah, it is important, and it's also good enough because clearly all the invertebrates are surviving now. They're doing good. They're doing very good. All the remember which is the biggest phylum? Class 11th. Biggest phylum in animal kingdom is the phylum Arthropoda. So if there is anything which is highest in number on the planet, it is insects. And insects do not have adaptive immunity. So they're doing pretty fine, right?
Yeah.
With just innate immunity. Sometimes it's not the complexity that matters. It's the efficiency of the simplest of the systems that matter in biology. Right? So that's why, and when a human is born, adaptive immunity is slow. It takes time to be formed. So if we do not have innate immunity right from the beginning, we are just open for any attack. Right? So it's very important to have innate immunity. Anyone has any doubts with innate immunity? I'll just stop here for a minute before I go into acquired.
Yes.
So yes, tell me.
I'm in no doubt. So.
Okay, cool. So whatever we studied in innate immunity, now we are going to build on that. Okay, so we have not studied antibodies, memory, anything like that. So this is all that we have by birth in our system to protect us, and it works really well. Now let's go to acquired immunity. Innate. Acquired immunity, or also known as adaptive immunity.
Adaptive.
Yeah. So it's both words mean something. So acquired means we acquire it after the birth, and adaptive, what does the word adaptive means? Why it's
We
called-
get used to it and develop a behavior or develop something.
we can adapt it, we can change it, we can rewrite it. That's what adaptive word means. Innate immunity is set in stone. If you have natural killer cells, you have natural killer cells. If you have macrophages, you have macrophages. What they do, they do. But in adaptive immunity, you can change the adaptive immunity with respect to the pathogen, the kind of pathogen you are being exposed to. Or if the same pathogen also changes and is constantly in nature, in a tussle, then you will also change your adaptive immunity with respect to the change in the pathogen. And it will happen eventually for COVID as well. It will just take time. And why humans are different? Because we always try to protect our kind. It's not the first pandemic. Pandemics have happened, always have happened. Even when we did not know about anything, we were just cavemen or Homo sapiens just came up. Then also there were many pandemics. No one documented it. Pandemics have to be there. Like pandemics will come. There is always, I think many scientists are also working on to predict when is the next pandemic going to happen and what will be the nature of that pandemic. Because it's not entirely impossible. We already know that the kind of activities humans are doing, the kind of effects we are having on environment, we are very prone to zoonotic jumps. Zoonotic jumps means some virus which used to be a pathogen for, let's say, pangolin, or let's say dogs or cats. Now, because of the interactions and because we are living in close proximities with them more and more, habitats are not segregated. We are more prone. The virus will get more exposure to different hosts, and the virus is more prone to jump or change or shift host. That's called zoonotic jump of a pathogen. So that's very, very common to happen in future as well. But the problem is we don't just handle it with pure science. There's lots of politics and other things involved. Society is not ready to accept many things. Before COVID, the world was neglecting viruses for a very long time. People were not studying viruses. There were a handful of virologists in India who are actually working on viruses and trying to understand. And some of them actually warned that, our negligence for the viruses will cost us if something happens, if any viral epidemic erupted or a pandemic erupted. And that happened. Now, all of a sudden, everyone is interested in studying viruses. And we are neglecting bacteria and protozoa for that matter. So you understand what's the problem, people? It's like a trend, So pandemics are also like a trend. It was like some reports also suggest that it was very visible from last 20-25 years. From last 2 decades, if you study the pockets of epidemics in the world, it was very suggestive that a big outbreak is going to happen somewhere and it will be of a virus. But now when we retrospectively read those reports, understand those, connect those dots, we are like, oh yes, there were signs. But when there were the signs, when there were red flags, no one saw it. When the world is suffering, now everyone can see that, oh, there were red flags. But this is human behavior, right? If I warn you that, okay, don't smoke cigarette, you will be like, okay. But if something happens due to smoking cigarette, then we're like, oh, someone warned me not to smoke cigarette. Correct? That's human behavior.
Yes, sir.
Yeah. Okay. Anyways, so about adaptive immunity, the most amazing thing is it remembers. Okay. It's like if anyone of you is a fan of Game of Thrones, they say the North remembers. So the adaptive immunity remembers. And how it remembers is, it seems to have a memory. a memory of the first encounter with the pathogen or any antigen to which you were exposed for the first time in your life. One good example can be COVID. Many of us got exposed to COVID for the first time in our life. Any one of you here has tested positive for COVID at any
Yes.
given time? Matthew, you have tested positive, right? And did you get symptoms, or was it?
Yeah, I had symptoms. Yeah.
Were it severe, mild?
Mild. It was not like severe.
No severe symptoms, right? No pulmonary issues, just
No, no,
fever,
no. Fever, cough, cold.
cough, cold, loss of taste and smell
No,
also?
no, not loss of taste, but fever, cough, and cold for sure.
Okay,
So-
which means you were not infected by the Omicron or the Kappa variant, because they were also adding to the loss of taste and smell, because they were killing the epithelial neurons of your olfactory system so that you could not smell, because these sensory neurons which bind to the smell molecule are dead. But the good thing about these neurons is they can regenerate. So after the infection is gone, they regenerate back and you can smell back. Okay. So just by looking at symptoms also, we can kind of see that what kind of strains you were infected from. But yes, now if you go and get yourself tested, your body, your blood will have antibodies against that particular strain of COVID from which you were infected. You know that? Did you get
Yeah,
an
yeah,
antibody
I can't.
test?
No, I haven't got antibiotics, but I'm like aware of this. I've
Yes, you
read
are aware.
about
So
it.
if you do antibody test, you will get to know that yes, your body has antibodies. Now, this is where Matthew's body now have a memory of this encounter in the form of these antibodies. Now, what are antibodies? So before going into it, let me tell you about what is a primary response and what is a secondary response. So acquired immunity, it has a memory component, which innate immunity did not have. There was no memory. They were just trained cells. By birth, they just needed to do one thing. Okay, if macrophage is there, it will see anything which is foreign and will try to kill it. It will not remember while doing that. Okay, I killed this bacteria which had this kind of a receptor on the surface, and I should remember if the next time the pathogen comes. No, but acquired immunity does that. So it has a memory component. Now there are two things. So the first time when there is first encounter with pathogen, and if this when we are studying acquired immunity people, we will assume all the time that whatever the infection was, the innate immunity was not enough to handle it. Okay, that's the baseline assumption. That's only when we can understand the adaptive immunity. In our body, both are working hand in hand. You cannot just tease out one from the other. Okay. But here, when I say first encounter, no one will ask me, or no one should ask me, why didn't our acid kill it? Why didn't the skin or the primary or cytokine barrier or cellular barrier kill it? Because we are assuming they could not. That's where it came to this point. And by this, I'm not saying that first the innate immunity works and then the adaptive immunity comes. I'm just saying that innate immunity is fast. It is ready-made. It's like an instant noodle. You're hungry, just put it in water, boil it and eat it. But adaptive immunity is like a biryani. It will take a long time. I give too much references for biryani, right? I have realized that. Anyways, so the first encounter with the pathogen is called a primary encounter. And against this, our adaptive immunity or acquired immunity shows a primary response. Now, what is a primary response? A primary response is like where our adaptive immunity send out a team for an investigation. Okay, they will go try to look at the pathogen, try to figure out what are the weaknesses of the pathogen, what the pathogen looks like, Which receptors does it recognize? How can we recognize it? What are the weak areas where we can bind or an antibody can bind? And how can we kill it? So all this analysis takes time. So primary response is always slow and has a low intensity. Make sense, everyone?
Yes, sir.
Slow to mount, like slow to begin, and it has a low intensity. But the benefit with this is, if you can manage with all the components of immunity that we have, if we can manage to defeat the pathogen, if the pathogen killed the person, it's gone. There is no need for a secondary response. The person is dead. But if the pathogen is not in very strong numbers or could not fulfill its motive clearly, and our primary response, though it was slow and had a low intensity, can get rid of the pathogen. Now, this point onwards, this primary response generated antibodies. Okay. Now, these antibodies, they're called antibodies because the word anti means anti.
Opposite.
Yes, against or opposite, right? So what are they against? What are they opposite of? Bodies. What bodies? There must be some.
Antigens.
Sorry.
Foreign
Antigen.
bodies.
Yes, foreign particles. Living or non-living, both can come into it, but foreign particles. So they are against some foreign particles. Now these antibodies, let's see what are they. So first write down. Okay, before you write down, let me complete. So this primary response generated antibodies. Now, if there's a second encounter, let's say you again got contaminated with the same pathogen. So second encounter with, I'm calling it pathogen alpha. Second encounter with the same pathogen, pathogen alpha. What will it lead to? It will lead to a secondary response. And this time, the body will not send an investigation team. What it will send? It will directly send a battalion. Okay? An army. Why? Why can a body do that? Because we already know how to kill the same pathogen, right? What are the weak points we have to attack? It's like you're boxing and two boxers met for the very first time in rings. Both of them do not know each other's tactics, do not know. So the first bout between them will be more of like investigatory. Have you seen two boxers like fighting for the first time? They try to stay away from each other for some time.
Yes.
For the first few minutes. Why are they away? They're not scared. That's a strategy. They want to know, they want to see the footwork, how agile, how fast the opponent is, what are the strengths.
Yeah, essentially like studying the opponent.
Exactly. They're studying the opponent. It takes time to study the opponent. Okay. So first bout, the first round, they always start like they're studying the opponent. They will land some blows here and there and they'll get some point. But you'll see the second and the third bout, if it goes to that, that's more vigorous. Because now they know, okay, this person always uses the left hand to mount the punch or is very good with the left foot, but the right is weak one. So let me slide to this and counter punch. So these things already have been made, right? So next time they both are well prepared. Okay, you understand?
Yes, sir.
Yes, sir.
The same thing is happening here. With the first encounter, we are preparing, we are mounting a response, we are trying to figure out the weaknesses. Second response is fast. It's a rapid response. And it is very high intensity response. So it's fast and it's like a high intensity, like there will be many millions and millions of antibodies We just pump it into the system. It's a war. Go and fight the war with this mindset, okay? So this kind of high intensified response is in biology is also known as anamnestic response. Anamnestic response. Vigorous and fast response. So the neat question, there was one neat question. Anamnestic response is associated with the secondary response of innate immune system, secondary response of adaptive immune system, primary response of adaptive immune system, and primary response of innate immune system.
Secondary response of adaptive immune
System. Innate immune system do not have a primary or a secondary response. All their responses are same line, same baseline, right? They don't learn.
Yes.
They don't have a memory component. They're not making strategies. They are. It's not like for the first time you encountered a pathogen in your stomach, your HCl destroyed it. The next time if the pathogen comes, you will start producing more HCl. No. You just know it just got destroyed because it came into the way of HCl. That's all. There's no memory component there. Make sense, everyone?
Yes, sir.
Fatima, Kulsum, and Matthew.
Yes, sir.
Okay. Now the primary and the secondary response are carried out with special types of lymphocytes again. So I told you the lymphocytes will come back. So for both primary and secondary response, the crucial thing is two special types of lymphocytes. Anyone knows? Now they are not.
Lymphocyte and T lymphocyte.
Yes, they are again lymphocytes, but remember lymphocytes was here also. Sorry, neutrophils, monocytes, and NKCs. These three leukocytes were there. The kind of leukocyte that you will see here, two special type of, these are lymphocytes. And they are called B lymphocyte. Anyone knows why is the name B and T? All books write it as B lymphocyte, T lymphocyte, but what is B and T?
No idea, sir.
Okay, it's the origin. B lymphocytes are produced from bone marrow. That's why B lymphocyte, okay? And T lymphocytes are produced from thymus. So they're called T lymphocytes, okay?
Okay, sir.
Now, out of these two, only B lymphocytes can produce antibodies. Okay. Then what does T lymphocyte do? T lymphocyte helps the B lymphocyte to produce antibodies. So both play a role in antibody production, but B plays a direct role in antibody production where it produces the antibody directly, and T lymphocytes play an indirect role by helping or stimulating the B lymphocyte to produce antibodies. Is it clear?
So in what way does it help again?
Yeah. In what way? Now that's a question. Like if you're asking the molecular mechanism, that's very, very in detail. But what I can tell you in brief is there are some very essential factors that have to be released for the B lymphocyte to know, because B lymphocyte, it's like, let's say the coach and the person. So you are a talented person, okay, Matthew? So let's say you play badminton and you are very good at smash. So every shot you think that smash is my strength, so let me play just smash. But then there has to be someone who should tell you that, okay, this is not the right time to play smash, okay?
I see.
save it for some time. When it's the right point, then place mash, because there has to be same logic is in biology also. There has to be something which can look at the scenario from outside, because when you are into that act, you will do what you are good at. B lymphocytes produce antibodies, so they will just produce antibodies. But T lymphocytes kind of regulate that antibody production when in terms of temporal, which means time-based, and intensity-based production. Okay.
Okay, so
So the smash was just an analogy. If you, does it help you to understand what kind of, so it's an interaction between these two.
so essentially the T lymphocyte tells the B lymphocytes when to produce antibodies.
Yes, and in what
Okay.
circumstances to produce what kind of antibodies.
All right, fair enough. All
Okay.
right.
Yeah, and also that's why T lymphocytes also called sometimes T helper cells, because they are helpers. And this is exactly the kind of cells which are targeted. So there are two ways to make a team weak. Either make the player injured, okay, like I make Matthew injured, so Matthew cannot play, so it's gone. Or I make the coach of Matthew injured, so that he does not get proper training now, and he does not know what to do when. Do you understand?
Yes.
So AIDS, the virus that causes AIDS, you know that virus? We are going to study it in detail.
The
HIV.
human immunodeficiency virus, HIV.
Yes, it attacks and kills the helper T cells, and thus weakens the immunity. Okay. Now,
So is there any other way to
is there any other way to?
to make the B lymphocyte active other than T lymphocyte?
Yes, there are many other stimulatory ways. There are drugs now which we have formed, which are called immunostimulants, that can make basically they are on the same concepts. You put these drugs.
So those are given when you have AIDS.
Condition. People are trying that, but it's not, because there is one more complexity with it. Let me complete the story for T lymphocyte. T lymphocytes help B lymphocyte to produce antibodies. That's their part-time job. They cannot be just be a morale boosting cells, no. Biology also have a function for them. So B lymphocytes, because they can produce antibodies directly, and these antibodies are in the blood all the time. So they provide blood-based immunity. It's also called humoral immunity or hemoral immunity. Hem means blood. So you call it humoral immunity or hemoral immunity. It's blood-based. And blood-based immunity is mainly regulated by B lymphocytes. But thymus, apart from helping B lymphocytes to produce antibodies, so when it is not coaching the B lymphocytes, what it is doing? T lymphocytes themselves are also, they have one more function. They mediate the cell. Sorry, they regulate the cell-mediated immunity. T lymphocytes regulate the cell-mediated immunity. Now, what do we mean by cell-mediated immunity? Is that this is the direct function of T lymphocytes. Okay. And this is a part-time function. Like this is also an important function. I should not say part-time, but cell-mediated immunity means apart from blood's ability to recognize pathogens and kill pathogens. So any pathogen that infects may some or the other point of time will come in the blood, right? In the system. So blood-based immunity, antibodies-based immunity will work there. But there is another kind of immunity which is called cell-mediated, where our own cells and tissues knows that which cells belong to our own body and which cells do not belong to our own body. That is called recognition, self-recognition. So T lymphocytes help the cells to recognize our own cells. This immunity is very important and crucial in terms of organ transplant. Have you seen that people who undergo organ transplant have always have this risk of transplant rejection, graft rejection?
Yes, sir.
What is that rejection? Who is rejecting what? Our body is rejecting that kidney that was transplanted in that person because that kidney came from some other person. But we did match the kidney. There's something called organ matching before we do that, right?
Yes, sir.
In biology, there is never 100% matching. You cannot do 100% matching. It's not possible. Unless and until you are harvesting the organs from your own clone, okay? Or from a monozygotic twin. In that cases, the chances of rejections are very, very less or almost no rejection. But in any other chance, there will always be chances of rejection, and that rejection happens because the moment we transplant any tissue from any other person to any other person's body, our cell-mediated immunity becomes active and tells the body, oh, this is not our own cell, so let's kill it. They don't know that that tissue has been put in our body for our own benefit, because that is not natural. In nature, no organism can ever take any other organism's organ and put it in its body and survive, right? So nature always works on this one principle of recognizing self and the non-self. Medical science is our own discovery and invention and achievement. So we have figured out a way that, okay, if I match these factors and put this organ into this person's body, maybe this person will survive. But then we have to silence the cell-mediated immunity. And for this, we give the patient immunosuppressants throughout the life. So that patient always have to take one tablet or something in the morning and the night that keeps its cell-mediated immunity silenced. Otherwise, the graft will be rejected. Make sense, everyone? So T lymphocytes mediates cell-mediated immunity. B lymphocytes mediates the blood-based immunity. T lymphocyte also helps the B lymphocyte to produce antibodies. So indirectly, they also help in antibody production. So I'll stop here today, and in the next class, which you will get to know on the group if it is happening tomorrow, I will be talking in detail about the antibody itself. What kind of antibodies are there? What do they do? We will talk about vaccine, vaccination, immunization, passive and active immunity, and we will also talk about allergy in the next class. So please read about allergy a little bit. Allergy is something different from infection or inflammation, and we'll cover it in the next class. So next class is going to be a little interesting because whatever I'll talk about, you all must have experienced or you have seen it in your life. Okay, so we'll try to finish immune system in the next class and then go to some major diseases. Basically, 2 are in your syllabus, cancer and AIDS. So I'll teach you in detail cancer and AIDS after that. Okay, everyone?
Yes, sir.
Okay, sir.
Okay, and I also put on the group the information for biotechnology class. If anyone wants to join that for the purpose of exams, you can do that. Okay, see you.
Thank you, sir.
Thank you, sir.
Thank you, sir. Bye.